loose cannon tied up somewhere before he says something he really regrets. And we get to start off with scintillating topics like the genetics of autoimmunity. So for the next half hour, we're going to look at gene pathways, going locus by locus. No, we're not going to do that. So what I'm going to try and do for the half hour is something very simple. I'm going to try to explain all of the hype and all of the discussions and all of the news stories you hear on a regular basis about the genetics of disease, so you can put it into context for the genetics of autoimmunity and neuroimmunology. So if you remember Neuro 101, now we're doing genetics. Neuro's easy. Genetics is hard to a degree because the nomenclature is a disaster. But let me give you the basic introduction to genetics. So if you get this, you get genetics. You ready? It's got nothing to do with cats. Here we go. Everyone paying attention? All right. This plus this plus this equals that. Right? Okay. It's genetics. If you leave this out, anyway, so it's genetics. Everyone got it? We're done. Actually, this is very instructive. So take a look at these two beautiful creatures. Now take a look at these two. So I'll flip. This one, no paternity question whatsoever. This one, I'm waiting for the DNA test to come back. We got two brown hair, brown-eyed, and then you get a blonde hair, blue eye. I was on call a lot before she was born. So I don't know. But the difference between them really goes to the heart of genetics, because I have one genetic code, my wife has one genetic code. We created two humans, but they are exquisitely different in a variety of ways. And it's in those differences that you understand genetics. So you have DNA, right? Everyone remembers the double helix, Watson, Crick. They are made up of bases, this code, and we're missing G is hidden there, ACTG. So these bases, those 4 nucleotides, they're sugars basically, those 4 nucleotides are responsible for everything that you're doing right now. And it's the sequence of them that makes the difference. They pair up these base pairs to form the double helix, and you get what are called codons. So every string of 3, so ACT, TTT, CGC, whatever, 3, that's a codon. And ultimately, that may encode an amino acid on a protein, a single amino acid on the protein. Genes, by definition, are the collection of codons that are responsible for an individual protein. Okay? So think of any protein name you know. What's a good one? Who knows the name of a protein here? Doug, do you know any proteins? So if you talk about muscular dystrophy and dystrophin, the gene dystrophin, there's a single protein linked to the gene. Now, here's some of the complexity. A single gene can give you different proteins because you can splice the codes. So if I have this whole long code that's a gene for a protein, but I only tend to read half of it and encode that, that's a different protein than the whole code. You can also rearrange things. I can take the first part of the gene and the last part and put them together. How do we create all the billions of different antibodies that you have in your body against influenza, against measles, against the fungus that's in a room? You don't have that many genes in the human. You've got about 30,000. There's almost that many pathogens in the world. So if you had a single gene for each antibody, you'd be left with nothing else but a bunch of antibodies. So the rearrangement of genes and the splicing of genes creates another level of complexity here. And then there are alleles. And I know the language is getting tough. But as humans, we all have certain characteristics that are basically the same. We all have a liver, we all have lungs, we all have a spleen, some of us have a heart, the rest are in Congress. The alleles are in reference to certain genes that have a basic structure that's the same, but little differences between them that create the differences in us. Okay? So the difference between my daughters, they're both human, But the difference between the blonde head and the brown head is an allele. They both have a gene for hair, but one has a gene for hair that happens to look blonde, and one has a gene for hair that happens to look brown. Same things with eyes and everything else. So those are the alleles. Chromosomes, as the genes form long strands, as the DNA strands, we have sets of chromosomes, 23 pairs, okay, so 46. Some of us have one extra or one less. And the chromosomes are what come from mom and dad. So 23 from mom, 23 from dad, and you get a unique human being. And then all of your genes, all of your chromosomes in total are your genome. So the Human Genome Project is to sequence from chromosome 1 all the way through the 22nd autosomal chromosome and then the sex chromosome, every gene in the human body. Now, after hearing about this, is it possible really to know the genome? No. We can sequence one person. So, for example, Watson had his genome sequenced. It was in the news. It was kind of fun. It was done down in Houston at part of one of the human genome sites, and they published his genome. But his genome is just his. He's going to have alleles that are different, right? Now it gets a little scary. So we've got the human genome. It was actually based on a consensus sequence from several humans put together. What if we picked a bunch of people who are about to get colon cancer or something, and we think that that's the normal gene versus not? So there are an amount of statistics that go into this to decide what is an allelic change versus what is the background. And I'm not going to go into any of the statistics today. You're welcome. So genetic variability. So we can clone a sheep, right? We can take Dolly, we can make a new one, and the genes are exactly the same. And then we can clone it again and again and again and again. Actually, why did you really have to clone a sheep? Can you tell the difference at the baseline between any of them? But you can scientifically, you can clone certain genomes and create exact replicas of that genome. We have it in humans. There are human clones, so twins. So anyone here who has or is an identical twin, the genomes are exactly the same. Twin studies are great for trying to understand genetic contributions to diseases, because if two individuals have the exact same genome and everything in life is genetic, their lives should be exactly the same. How many people here know any identical twins? Are their lives exactly the same? No. They're completely different at every level. Personalities, diseases, issues. So we know right off the bat that life is not just genetics, it's the interaction of the two, but it's a useful tool for trying to sort out what is the contribution of genetics to a certain disease. So this is to remind you of alleles. So you have a genome, but you can have alleles with multiple different eye colors, and these days you can get contact lenses to just mess with this more. But all of these, each one of these humans has an eye. The eye is functioning. They see the same way. They have cones. They have rods. They have photoreceptors. The difference in their eyes is only the color, and that is an allelic change. The genes that go into the eyes are basically the same. The gene for eye color is basically the same, except for a few of those codons. Just a few of those codons are different, and then you get this variability in terms of eye color. Does that make sense? Kinda. We'll come back to this more. So alleles are known variations, the spice of life that make us all a little different on top of a background. Then you can have mutations on top of that. Okay? So these are things that shouldn't happen where the gene isn't functioning the proper way anymore. Here, the genes are functioning properly. They're just giving you a little bit of difference. You can have mutations where the genes just aren't working the way they're supposed to. And you get something like that. Oh, no, I'm sorry. I got to be careful. I figured I was in Washington State. I could get away with putting Dick Cheney up. Anyway. And then there's a whole host of genetic conditions. So normal variations you consider a genetic condition. So blue-eyed folks have the blue eye syndrome, the genetic condition. These are normal. There's nothing wrong with it. Then you can get into recessive diseases. So these are mutations, but they're mutations in genes where you have to get both mom's and dad's genome having the mutation. Meaning if either one of the proteins is working correctly, you're going to be fine. But if you get a mutation from mom and you get a mutation from dad, you're in trouble. Cystic fibrosis, Tay-Sachs disease. So cystic fibrosis, about 1 in 40, 1 in 50 people are carriers for a mutated cystic fibrosis gene. Do 1 in 40 people have cystic fibrosis? No. You have to have 2 people who are carriers, have a child, and then in that 50-50 Powerball, in terms of recreation, the child has to get the mutated gene from each parent in order to have a recessive disease. There are dominant diseases where if you have the gene, you have the disease. Huntington's is an example of an autosomal dominant disease, meaning if your parent has Huntington's, there's a 50-50 chance you're going to get it because it's only tied to the genome you get from that parent, from dad, for example. So if it just happens during the Powerball of recreation, you got that set of chromosomes from dad that happen to have that mutation, you're going to get the disease. So you can test for this, you can screen for an autosomal dominant, and if it comes back positive, you're going to have the condition. You can screen for recessive conditions, and if you have 2 copies of that gene, you're going to have the condition. And this is true 99% of the time. Few exceptions to that rule. Does that make sense? Are there any of the immune diseases, autoimmunity, that are recessive or dominant? We'll find out. Most of genetics and most of what you hear about on the news are predispositions. You inherit a certain gene, and you have a predisposition to a disease. So the BRCA1 gene for breast cancer, have you heard of this? BRCA1. So this is a gene found mostly in Ashkenazi Jewish populations, but it's throughout the world. And if you inherit this gene, your risk of breast cancer goes up significantly. But there are a lot of people with this gene, or the alteration in the gene, who never get breast cancer. This is a predisposition, meaning the gene does not dictate in any way, shape, or form with 100% accuracy whether or not you get the disorder. We've looked at predispositions for a long time. Okay, so you'll hear news reports all the time. I found the gene linked to being a moron. I found the gene linked to being a Yankee fan. This is far more prevalent than we'd like to admit in America. Genes linked to bad driving. Genetic risk of flatulence. These are your tax dollars going to these studies. I'd write your congressman. These are all made up. None of this is real. But you read this report, right? You're going into work, you're listening to NPR, and it's, a researcher at University of Washington, a researcher at Harvard has discovered the genetic link for fill in the blank. Autism, lung cancer, multiple sclerosis, all these things. And everyone says, yes, we got the gene. No, we got the genes that may augment risk to some degree. Now, if that augmentation of risk is 0.0001%, have we cracked the case? No, it's leading us the right direction. And then there are gene-environment interactions, and this is what I love the best, actually. I think this is the fun part. You have genes in your body that are different, that have allelic changes amongst us, or even some mutations, that their activation is completely dependent on the environment. Meaning, based on your exposure, they may or may not activate. This is an example from flies, from Drosophila, where if they have a mutated gene, they look normal until you put them in a hot or cold situation, and then they grow a second set of wings. All of the flies have that same mutated gene. They're never going to get a second set of wings unless you change their environment. Okay? So a genome in no way absolutely predicts what's going to happen to you as a human. The outcome for humanity is based on the genetic-environment interaction. The genes predisposed this fly to grow a second set of wings. He would have never grown the second set of wings if it hadn't been for the genes, but it didn't dictate that he was going to be a 2-winged fly. Does that make sense? It's kind of freaky, too. So do we see this in neuroimmunologic diseases, this genetic-environment interaction? The answer is yes. You can look at animal models. So Doug and I, over the past, have looked at a virus called Symbis virus and spent a lot of time with this. And if you take mice with different genomes, this is going to be the only sciencey slide you see, I promise. If you take mice with different genomes, so one genome's listed here and one genome's listed here, and you infect them both with the exact same virus, okay? These mice all die, dead. These mice can survive. Same virus, different host. The host genome dictated survival, dictated the outcome, dictated the symptoms that the mouse had during the infection. Do we see this in humans? And the answer is yes. Anyone here heard of West Nile virus? Yeah, you all heard of West Nile. So West Nile virus, the epidemiology of West Nile virus is a great study of host infection interactions. So when we did seroprevalence studies for West Nile virus, it turns out that 80% of humans who get infected with the exact same virus. This isn't viral mutations. This isn't one virus doing one thing and another doing another. This is the exact same virus. 80% of people are asymptomatic. You never know it. 20% of people get a fever. It's a funny fever. It doesn't go away. You might have a rash. It tends to hang around, and you might get diagnosed with West Nile fever. But what did we hear about in the news for the last 9 years? Encephalitis, right? We heard about all these people, this outbreak, this scourge. It started in New York City. That should tell you something. It spread coast to coast, and people were dying left and right of encephalitis, right? That's what we heard about. That's less than 1% of the people who are exposed to the exact same virus. So the outcome of these interactions are dependent a lot on the host as well as the exposure. Does that make sense? What about the genetics of autoimmunity? So are there autosomal dominant autoimmune diseases? Meaning if you get the gene, you get the disease. The answer is yes. These are extraordinarily rare. How many people here have heard of autoimmune lymphoproliferative syndrome? Yeah, we'll have that conference next week. Polyglandular autoimmune syndrome type 1, which is more of a recessive. I should have put in the recessive here. But there are autosomal dominant and autosomal recessive autoimmune conditions. These are extraordinarily rare. The majority of autoimmune conditions are based on predispositions. This is just a list of some of the conditions that have found genetic links, genes or versions of genes that place you at a higher risk. So, for example, the best known, ankylosing spondylitis, if you have a certain HLA, there's a certain gene within your immune system, you have a relative risk of greater than 150, meaning you're 150 times more likely to get this disease than people without that gene. It's not 100% of people get it, you're just much more likely. Now let's go down to multiple sclerosis. This is multiple sclerosis here. And if you have a certain version of an HLA gene, you have 12 times the risk. That number sounds big. It's actually quite small. This is a big number, 150. Once you get into single and low double digits, there are tons of people in this room who have these alterations who are never going to have these diseases whatsoever. This isn't the whole story. You have to have the right gene, the right environment, and a constellation of symptoms, a constellation of situations to get this to have an effect. So I'll spend time talking about how many people here heard in the last year, big paper, New England Journal of Medicine out of California, Harvard, Duke, an international consortium, about the genetics of multiple sclerosis. Lots of press about finding the genes linked to MS. People hear about this? Did it make news? Okay. So we started getting lots of phone calls and lots of questions. Well, does this do it? Do we understand the genes that cause MS? And it was a great study on multiple levels. It was massive. There were about 10,000 people in this study, families where you had parents and children where somebody was affected by MS. 10,000 subjects with data collected where they did analysis of different alleles over the whole genome. This took tens of millions of dollars, a huge commitment. And we found out that 3 genes that had been previously linked to MS are still really linked to MS. It's a necessary, very well done study, but it tells us a couple things. One is, those types of studies are only going to get us part of the way. And what's the reason? Well, first, we're lumpers. What do I mean by that? We still take anybody who gets the diagnosis of multiple sclerosis and we lump them together. And let me just tell you, after spending the evening looking over all your medical records and MRIs, there's nothing lumpy about this. When looking over 10 MRIs of somebody diagnosed with transverse myelitis, there's easily 5 different patterns there, 2, 2, 2, and 2. And yet, in a genetic study, I'm going to lump you all together. Does that make any sense? No, that's silly. I'm going to miss things. So if you can better define your population, the associations are going to be better. If I can find a way to separate out type A multiple sclerosis, type B MS, type A transverse myelitis, type B transverse myelitis, then these studies have a shot of really getting better numbers involved. The other issue is it only gets you part of the way. We are going to miss critical pathways with these large studies because those pathways don't rise to the surface statistically, but when you put those pathways in certain environments, they are critical. Okay? So, for example, when you look at MS, we hosted, I'm not sure if Art's here, we hosted a meeting at Cold Spring Harbor this past year on the genetic environment interactions of demyelinating disease. And so the Cold Spring Harbor, what they'll do is they'll take 25, 30 clinicians and scientists, they lock you in a room for 3 days, they feed you very well, lots of wine, it's very nice, but they say you can't leave until you answer this question. And the question that was biostatisticians, geneticists, environmentalists, nutritionists, neurologists in a room, locked in a room, and they were told, design the study to sort out the genetic environment interactions of demyelinating disease. Day 1, everyone's all excited, everyone's working, giving lectures, talking, working in small groups back and forth. Day 2, a couple fistfights break out, you're an idiot, break them up. And then day 3, we had to come back together for a consensus. Here was the consensus. There are 2 ways to approach these studies. There is what you really want, the Cadillac of studies to get this done, and then there are practical issues. So what's the Cadillac of studies to get this done? Cadillac of studies is anyone here familiar with the Framingham Heart Study? So the Framingham Heart Study, they took a huge population of people and they followed them prospectively. Agnostically, they just said, we're going to collect data about you and see what happens, and it was to look for heart disease issues. What we need to do is, and there's work on this, is the prospective study starting at birth. 100, 200, 500,000 children. Annual visits all the way through, say, age 30, with data and blood being collected on all of them, and then you get to study what were the predictors of outcomes. And you have to be open-minded about what you're going to collect. So if you're worried about living near power lines as a risk for something, you got to record when they live near a power line. If you're worried about Epstein-Barr, you got to test them on a regular basis for Epstein-Barr. If you're worried about who's worried, what did we hear? Vaccines. You got to track vaccines. Whether or not you ate the salmon or the cod, you got to track that. It's a very labor-intensive study. That's the way you're going to ultimately answer this. Is it going to be done? Probably not. There is the children's study, which is an NIH-sponsored study of 100,000 children from age 0 to 18 that has been in the works for years and that they're trying to get off the ground. We're waiting for it to happen, and we're trying to make sure they design it appropriately to capture all of this data. How is that going to help us, though, if we have diseases that aren't going to present till 30 or 40? I need those kids to be followed past the age of 18. And so we've been petitioning, we've been talking with them, how do you tack on studies after the event? So if you can't do the Cadillac of studies, what's the practical one? So we've been designing this and working on this for neuroimmunologic diseases. We started with MS as a model, and we're working back from there. And the idea is, could you enrich the population that you're studying for people who are going to have a high conversion rate? Because if transverse myelitis has an incidence of, and we can start a fight about this, 5 in a million to 20 in a million, depending on how you define things, I've got to follow 1 million people to catch 5 cases. Does that make sense? It's a big, big study. But if I take the siblings of children with multiple sclerosis, or the children of parents with multiple sclerosis, where the rate goes to 2 to 5%, for every 100 I follow, I capture 2 cases. So the studies that are being designed now, and this is part of the work that the Accelerated Cure Project is doing, is to say, all right, let's design practical studies that we can do to try and start chipping away at the surface of this. Because in the end, you have to remember that fly. You have to remember the fact that there are going to be genes amongst us that may never come to play unless you get the right exposure. And for all of these disorders, transverse myelitis, neuromyelitis optica, ADEM, optic neuritis, it's that combination that's key. So in the end, the genetics of autoimmunity is extraordinarily complex. Could I give you a lecture on each of these genes? What is the significance of the interleukin-2 receptor? We can. I'm not sure you want to hear that, because even after I spend an hour describing what it does, it's a great story for why it could be implicated in autoimmunity, but it's never going to be the whole story. What we really need to focus on is how do we get to the whole story, and it's by making sure that we don't make assumptions, that we design things in prospective studies in such a way that we gather that gene-environment interaction. So the next time you hear on the news a study about, we found the gene for fill in the blank, you want to pay attention to the wording. Is it predisposition, or is it the gene that causes it? One of those autosomal dominant conditions. Those have become extraordinarily rare. We've found most of those, if not all of those. We're left with these, trying to chip away at the predispositions. So I'm on time this morning.